to the next installment of the magical molecular world. In the previous session, while winding up, I left you guys with a question that the DNA polymerase, if incapable of initiating the replication process, then who initiates the replication process and why? Now, why is very much clear in the first video itself, because I have already said that DNA polymerase requires 3' OH end for its activity. Now, when the DNA needs to be synthesized from the root, there is no 3' OH end available for the DNA polymerase to carry out its polymerization activity. So therefore, there needs to be someone who help the DNA polymerase by providing the 3' OH end. And this help is facilitated by a RNA molecule that we term it as primer. It is this primer that can initiate replication process. Now, what do we mean by initiating the replication process? Now, initiating the replication process means adding the initial few complementary nucleotides in accordance with the parental strand. Now, while adding the first few nucleotides, the RNA molecule provides the 3' OH end that is required by the DNA polymerase to carry forward its activity, and thereby the DNA polymerase plays the prominent role of polymerizing the newly synthesized DNA. Now, having known that, let us look at the different categories of DNA polymerases. Though in this video we will be very much specific and stick our discussion on the DNA replication process that happens or occurs in prokaryotes, especially in eubacteria, where we have taken a representative, that is E. coli, which will help us to understand the replication process in the prokaryotes or the eubacteria. Just as an overview, I would like to state the different classification of DNA polymerases and their basis of classification. So basically, DNA polymerases are classified into 7 different families ranging from A, B, C, D, X, Y and RT. This is 1, 2, 3, 4, 5, 6 and RT is a single one, being the 7th. Now, these 7 families of DNA polymerases can be classified as template dependent DNA polymerases, which are mostly common, and the second one is template independent DNA polymerases. Now, here we need to understand what the template means. Now, DNA polymerase during its activity requires a reference point, and it is the parental DNA strand, that is one of the parental DNA strand, which provides a reference point for the activity of DNA polymerases. That means by looking at the nucleotide combination present in the parental DNA strand, the DNA polymerase goes on adding new nucleotides, thereby providing the newly synthesized DNA strand. Now, the parental strand, which acts as the reference point for the DNA polymerase to actively perform its function of polymerizing the newly synthesized strand of DNA, that particular parental strand is known as the template strand. So most of the DNA polymerases are template dependent, and a very few, there is DNA polymerase which is template independent. One of the example of DNA polymerase which is template independent is the terminal transferase. Alright. Now, the template dependent DNA polymerase can be again classified into 2. Now, those DNA polymerase that uses DNA as the template, they are known as DNA dependent DNA polymerase, because they are using DNA as a reference point for its activity. So therefore it is known as DNA dependent DNA polymerase. On the other hand, there are certain DNA polymerases which uses RNA as the template. Therefore, it is known as RNA dependent DNA polymerase. An example of DNA polymerase that uses RNA as a template is reverse transcriptase. Reverse transcriptase. Transcriptase. Now, it is for a discovery of this enzyme Tamarind and Baltimore got a Nobel Prize. This is an extra information which I am giving to you. Now, those DNA polymerase that uses DNA as a template can again be classified into two. That is, those DNA polymerase which actively perform its activity in prokaryotes, and on the other hand, there are DNA polymerases which perform its activity in case of eukaryotes. Now, let's look at the prokaryotes first. There are 5 different types of DNA polymerases ranging from polymerase 1, DNA polymerase 2, DNA polymerase 3, DNA polymerase 4 and DNA polymerase 5 that actively plays a role in the replicatory process in prokaryotes. There is an E. coli, which is an example of eubacteria. Now, each and every different categories of DNA polymerase that actively plays a role in the prokaryotes will be discussed later in the video. Now, for now, let's look at the eukaryotic DNA polymerases. There are more than 10 different types of DNA polymerase that actively plays a role in eukaryotes. Now looking at the nuclear DNA polymerase, there is approximately 10 types of DNA polymerase. Now, as we have understood what is nuclear DNA polymerase, nuclear DNA polymerases are those DNA polymerases which plays an active role in replicating the chromosomal DNA which is present in the nucleus. On the other hand, there is also DNA as mentioned earlier in the extranuclear region, for example in the mitochondria. So therefore, for the replication of such DNA, there is mitochondrial DNA polymerase present, and one of the example of mitochondrial DNA polymerase is polymerase gamma. Now, in eukaryotes, we will be discussing about polymerase alpha, polymerase delta and polymerase epsilon, as these three plays a very vital role in the replication process of eukaryotes. Okay, having said that, let's stick to the replication process in the eubacteria, that is in E. coli. Now see, this is the representative which we have considered, that is E. coli. Now, one of the DNA polymerase which we have studied as one of the important type of DNA polymerase that plays an active role in E. coli is DNA polymerase 1. Let's look at some of its specification. Now, DNA polymerase 1 is also known as the Kornberg enzyme after its discoverer known as Arthur Kornberg. Now, basically, DNA polymerase 1 is a monomeric protein with polymerase as well as exonuclease activity. Now, polymerase activity, we know it very clearly that it helps in polymerization of the newly synthesized DNA, and exonuclease activity is this DNA, which is a monomeric protein, has got a trimming activity. Now, why this trimming activity is required? Now, trimming activity or the exonuclease activity is required for two different reasons. One, either the primer which initiated the DNA replication needs to be removed, or there is some sort of error which is present during the replication process and that needs to be corrected immediately. Then also the exonuclease is required. So the DNA polymerase 1 acts both as polymerase as well as exonuclease. Now, when a particular experiment was performed, that is when DNA polymerase 1 was digested with trypsin, this monomeric protein gave us two different segments. One segment contained the amino terminal and the other segment contained the carboxy terminal. The segment that contains the amino terminal is a shorter segment, and the segment which contains the carboxy terminal is a relatively longer segment. The longer segment is also known as the clino fragment after its discoverer. Now, since it is a protein, it will have an amino terminal and a carboxy terminal. So the smaller fragment contains the amino terminal, and the larger fragment, that is comparatively larger fragment, contains the carboxy terminal. Now, this two fragments perform two different activities. When observed, the smaller fragment performed 5' to 3' exonuclease activity. It performed 5' to 3' exonuclease activity. And do remember, whenever exonuclease activity is performed from 5' to 3' direction, it is always for the primer removal. As I have already said, that exonuclease activity may help in primer removal as well as help in error correction, but the small fragment helped in the primer removal by performing the exonuclease activity from 5' to 3' end. That is, it performed RNase activity. Now, as I have already said, that the primer is basically a RNA molecule. Now, since this RNA molecule is removed with the help of the smaller fragment also performs RNase activity. Now, coming to the larger fragment, it has got two different functions. First function, it performs 5' to 3' polymerase activity, and the second function is it performs 3' to 5' exonuclease activity. Okay, looking at some other specification, the polymerization rate is approximately 20 nucleotides per second. That is, in per second, the DNA polymerase 1 can add up to 20 nucleotides, and its processivity value is approximately 200 nucleotides. That means the DNA polymerase 1 can add up 200 nucleotides at a time. Okay, now having understood about DNA polymerase 1, let's look at DNA polymerase 2. Now, DNA polymerase 2 is again a monomeric protein with a polymerization rate of approximately 40 nucleotides, which is slight higher than the DNA polymerase 1, where in DNA polymerase 1 the polymerization rate was approximately 20 nucleotides per second, but in DNA polymerase 2 the polymerization rate is approximately 40 nucleotides per second, and its processivity value is approximately 1500 nucleotides. That is, at a time DNA polymerase 2 at a time the DNA polymerase 2 can add up to 1500 nucleotides. Now, looking at other functions of DNA polymerase 2, then DNA polymerase 2 performs 5' to 3' polymerization, 3' to 5' exonuclease activity, and this exonuclease activity, which occurs from 3' to 5' direction, is always for proofreading or error correction, and the exonuclease activity that occurs from 5' to 3' direction is always for primer removal, as it is the case in DNA polymerase 1. Now, DNA polymerase 2 involves in DNA repair, obviously, because it has got exonuclease activity, and it can initiate the DNA replication from a completely new site. That is, for example, if there is any sort of error in the middle of the newly synthesized DNA, then it can trim that particular error part and can initiate the replication process from there within. Now, coming to DNA polymerase 3, which is the most prominent enzyme working actively in case of prokaryotic DNA replication process, is DNA polymerase 3. Basically, it's a multimeric enzyme. That is, it is made up of more than one unit. That is, it is made up of 10 subunits. It is also known as the DNA replicase, as it is the main enzyme participating in the DNA replication process of the U bacteria, that is in E. coli. The polymerization rate is the highest from the other DNA polymerases actively working in the U bacteria. That is, the polymerization rate is approximately 1000 nucleotides per second, and processivity value is approximately 5000 nucleotides. Now, performs 5' to 3' polymerization and 3' to 5' exonuclease activity, and this 3' to 5' exonuclease activity is basically for proofreading. And this enzyme, which is DNA polymerase 3, is also known as the holoenzyme, because this enzyme, the different components of this enzyme, they work together as a whole to execute a single and the most prominent process, that is DNA replication. Now, if you want to look at the schematic diagram of DNA polymerase 3, then it looks somewhat like this. Now, it has got 10 subunits, out of which it has got 2 pairs of catalytic unit. Now, this catalytic unit is basically the main unit which performs the actual function of the DNA polymerase. That is, it performs the polymerization as well as the exonuclease activity. Now this catalytic core is associated with the clamp loader, and this catalytic core is associated with the clamp loader with the help of a dimerization component. We have got a pair of dimerization component, as you can see, but I have only struck it at one side of the holoenzyme. You can see the similar structure in the other side. Okay, so I will show it in one side, that is, it has got one pair of catalytic core, it has got one pair of dimerization component which helps connecting the catalytic core with the clamp loader. It has got a pair of beta sliding clamp. Okay, now having said its different component parts, let us look at its function. Starting with the catalytic core, now catalytic core can be categorized into three subunits. One is alpha, epsilon, and the theta. The alpha subunit of the catalytic core, it involves in 5' to 3' polymerization. Epsilon involves in 3' to 5' exonuclease activity, which is required for the proofreading process, and the theta enhances the activity of epsilon. That means whatever activity epsilon performs, that is proofreading activity, that is enhanced by this theta component of the catalytic core. Now coming to the sliding, now see, dimerization component, is very much understandable that this dimerization component helps connecting the catalytic core with the clamp loader. I will distinguish between the activities of beta sliding clamp and clamp loader, but before that you need to understand one thing, one very basic thing, that whenever DNA polymerase tries to perform the polymerization activity, there is always a tendency for a DNA polymerase to get separated from the parental strand, thereby halting the DNA replication process. So in order to inhibit this particular scenario and helping the proper synthesis of the DNA strand, there is a clamp loader which helps the DNA to stick to the parental strand via this sliding clamp. That is, this beta sliding clamp, which we have in pair in terms of DNA polymerase 3, this slides along the parental DNA strand, which is acting as a template strand. Now this, though help in sliding of the DNA polymerase in the template strand, prohibiting its separation from the template strand, but it is always the clamp loader which helps loading the sliding clamp into or within the parental strand, thereby executing the process of DNA replication smoothly. So that's all for today. Here in this video we have learned about different types of DNA polymerases and their different functions, where we have also learned that which is the major DNA polymerase acting in DNA replication in terms of eubacteria, more specifically in E. coli. And we have also discussed the various component parts as well as its function of DNA polymerase 3, which is also known as the DNA replicase, being the major enzyme. So in the next video we will dig deep more about this wonderful replicatory process taking place in E. coli. So that's all for today. And at the last but not the least, I always ask my viewers that please do subscribe. But from now, I won't ask you to subscribe, but just look at the videos that I'm uploading. And if my videos help you an inch, then I will think that my purpose for creating the channel is fulfilled. So that's all for today. Thank you very much. Let's wrap the video.